Okay, in this video we're going to look at materials moving across that cell membrane, that selectively permeable phospholipid membrane because of concentration differences. Basically, you can see here things move from a high concentration to a low concentration from here outside to inside. And we'll take a look at the forces that might be at play and give them some names and explain why this happens and some applications for this. This type of transport we call passive transport because there's no energy required from a cell. The cell doesn't have to expend any ATP to do this. Now energy is behind it, but it's not energy the cell has to use. Now molecules move across the cell membrane this way, but not all molecules do. They're only going to be able to move across this way if they have a transport protein that they can move through, or if they're dissolvable in that cell membrane, and they will always move from a high concentration to a low concentration, and we call that concept the concentration gradient. This is a big concept. We'll come back to the idea of concentration gradients over and over again, especially when we do cell respiration and photosynthesis. Now diffusion is any molecule. We'll do this, but a special case of diffusion will be osmosis, and this is the diffusion of water. When water diffuses, we tend to call the term osmosis, and every substance moves along its own concentration gradient. Now this is sort of the textbook version of what we look at with diffusion, where we've got a high concentration of something like a drop of food coloring or dye, and then we give it some time and it spreads out throughout the whole liquid, throughout the whole beaker, the whole glass. That's true, it works, but it's not quite as simple as stuff moving from where there's a high concentration to it to a low concentration. Now what's really going on is these molecules are randomly moving, usually because of the energy in their systems of heat, and they basically move randomly, but of course the more they randomly move, the more likely some of these molecules are to find their way into places they haven't yet been, and we can sort of show that with a little animation. Now what's happened with this video is they've taken a single particle and highlighted it here in yellow, and it's going to trace itself as it moves around. Now this set of molecules is already pretty well diffused, but if you watch this thing how it goes, you can see that it's moving pretty well as it goes through here on its own. So there we go. And you can see it's moving around here all over the place. It's making collisions. Its movement is pretty random. It's just determined by what molecules it collides into. Now obviously if you can imagine this happening in a closed space, these molecules are going to bang into each other a lot, and they're going to end up careening off into the open spaces where there's not a whole lot of molecules, just by pure chance. That's really what's going on in diffusion, just random motion of particles. So even when something's going from a high concentration to a low concentration, like we saw in that picture with the beakers, what's really happening is there's just as much movement each way, it's just there's more particles to move one way than the other, and we talk about it being a net movement. And the motion is really, really random, and there's lots of great animations out there and models for this sort of stuff that you can take a look at. Now there's three types of solutions in terms of concentrations we would worry about. And the first one is isotonic, and this is when the movement of particles in is about the same as the particles going out. So your concentrations of solute in the solvent are the same on either side of the cell, whether you're inside the cell or outside the cell. If it's a hypertonic solution, basically what's going to happen is you've got more concentration of solute, okay, the dissolved stuff, outside the cell than inside, so you're going to get a net movement usually of water, especially in living systems. You're going to see that net movement of water coming out of the cell in hypertonic solutions. And what that means is our cell is going to end up sort of taking this crinkly sort of shape on, because as the cell membrane shrinks down. If it's a hypotonic solution, what's going to end up happening here is our net movement of water is going to flood into the cell, and that's going to cause our cell to really expand. And if it expands too much, just like your bubbles yesterday, your cell membrane may burst, and you're going to have that cell be exploded. This is the reason behind why your grandmother, your mom, or your aunt might have told you it's a good idea to gargle with salt water, because that's a very hypertonic solution. The salt causes the crinkling or crenation of those bacterial cells in your throat, and they're not able to deal with that change in osmotic pressure and in water concentration very well. They don't have the structures to deal with that, and it often kills them. That's probably why if you had your wisdom teeth out, your dentist wanted you to gargle and rinse with salt water for a couple of weeks after anyways. It's just a nice visual in terms of what we see with red blood cells. It illustrates it very nicely. We've got the isotonic solution where our solution's moving in and out. The hypertonic solution where we've got more water moving out of our cell because we've got a high concentration of solutes outside the cell. And our hypotonic solution where we've got a large concentration of solutes inside the cell, so water rushes in, causing that cell to expand. And these are all human red blood cells, or mammalian red blood cells anyways. Now, not everything can get through that cell membrane perfectly. A lot of things, especially if they're charged particles or they're polar or just simply very, very large, have to come through channels. And if we remember our cell membrane, we've got those, jellyfish-like structures. And then every so often you've got a large transmembrane or across the membrane protein, and that acts like a pore. Now if it's something like water, it's going to be an aquaporin, which allows water to go through, and the shape of that protein, the hole in it, is specific for water molecules, and it will just allow water to go through. There's other proteins, some of them have to be unlocked. Some of them require another molecule to bind to it, and that concept where things get across the membrane by sort of being helped or walked across, like helping an old lady across the street, we call that facilitated diffusion. It's still passive transport. It happens from a high concentration to a low concentration, but there's no energy required, and it happens through one of these transmembrane proteins, these gateways, these doorways. And if you remember our cell membrane bubble lab, that was that thread ring that you could move around on the surface of that membrane, and that's exactly what these transmembrane proteins do. They're not set in the cell, they're all moving all around, and as they come into contact with materials outside the cell, they can rush into the cell or vice versa, and that's how a lot of materials are moved in and out of the cell to maintain concentration gradients. But again, those facilitated diffusion proteins, these transmembrane proteins, they can be opened or closed as the cell needs that to happen.